And now we should be on YouTube for FSN328. Let's see. Last time we went through this, right? Talked about kind of the structure of the nucleus, and we said what each of those different items up there was about. We talked about this slide as well, right? Mm-hmm. And one of the things that I ended up with last time was I said, you know, out of all of those potential genes, and I don't remember if I gave you a number or not, you guys know, for example, how many potential genes they think there are? Did I give you that number? You've probably heard this, right? When they start talking about the Human Genome Project and they said, we're done. And everybody went, so what does that mean? Well, one of the points of the Human Genome Project was now they had the sequence of all of the nucleotides of the human genome. Then the next piece of work was, let's find the genes. Why? Because the genes make the proteins, and the proteins are the molecules inside cells that give them specialized function, right? So one of the points of this was they said, you know, based on this information and what we know about it, we're thinking there's about 30 or 35,000 different genes in the human genome, which means there's potentially 30 to 35,000 different proteins in our body. That seemed like a lot. 30 to 35,000. I don't know. Must be adequate. Will we ever know what all those proteins do? No. But some of them are related to nutrition, so we're going to focus on some of those. And one of the things that I drew up on the board last time is I said, out of all of those genes, only a small amount of these bases are part of the genes. So we said there's about 4 to 4.5 billion different nucleotides. And of all those nucleotides within that, oops, I think I'll draw right here. I said, let's say, for example, these are the nucleotides of a single chromosome. And let's say this is 1 million nucleotides long. And if we were to find the genes in this, it might be a small little piece right here. Might be a small little part right here, maybe here. So if we focus on this, one of the questions is, how does the cell know that this is a gene that needs to be transcribed? And one of the things we said was, how are genes identified and expressed? Well, it's done by proteins. Proteins are the molecules that are actually looking for genes. The types of proteins are sometimes generically referred to as transcription factors, and some of them are enzymes. They find specific binding sequences to bind to, and then transcription can be initiated. So the proteins are looking for specific nucleotide sequences. Another name for those nucleotide sequences are promoters. And I said in here, promoters are nouns. So if you're going to define a promoter in here, you could say a relatively short segment of DNA. A relatively short segment of DNA that regulates the expression of a gene. Okay? So we don't say this protein is promoting the expression of a gene. We say the promoter is a relatively short segment of DNA that controls or regulates the expression of a gene. So it's DNA. Typically, for the most part, promoters are upstream or in front of genes, and promoters are targets for protein and enzyme binding, so they help promote the expression of genes. Little review here, right? I'm going to give you pieces of this and then I'll kind of put it all together. DNA to RNA is transcription, RNA to protein translation. Overall, sometimes that's referred to as gene expression. We're expressing a gene and making a protein from that transcript. Remember, the DNA is unique to each person, although the DNA in each of that person's cells is theoretically the same. But our DNA, everyone in here's DNA is slightly different. The reason that makes this different is because that causes different proteins to be made. The proteins are slightly different. That's what gives us certain physical differences, psychological differences, nutritional differences. All of that, most of it is because of the proteins. So that can lead to possible differences between people. Okay, so we know transcription involves going from DNA to RNA. So let's review real quick what are the differences between DNA and RNA. We know DNA is double-stranded, right? RNA is single-stranded. It's a single-stranded nucleic acid. Which one of those tends to be more stable? DNA. We've all seen Jurassic Park, right? This is kind of interesting. I'll tell you a side story because this is related. When I was an undergrad at Cal Poly and I was working in a lab, one of my advisors came into the lab, was talking to another person, and then they talked for a few minutes about things. I didn't understand any of it. He walked out. My professor said, "Do you know who that is?" And I said, "No." She said, "That's Dr. Raul Cano. He's actually, I think, just retired from here." And I said, "Oh, really?" And she said, he's doing some really interesting research. And at the time, I was working on immunology stuff. And she said, "He's actually doing some DNA sequencing." I said, "Okay." He's taking blood samples from mosquitoes, extracting the DNA and sequencing that. And he's trying to characterize, figure out where that DNA actually came from. I went, "Oh, that's really interesting." Not really. Just got to finish my thing here, right? I remember thinking, I don't know what that means. So flash forward, I don't know, 5 or 10 years, I'm sitting in the first part of Jurassic Park, that movie, I'm going, "Are you kidding me?" That's how they started doing it, right? Investigators like that were actually able to find that. So is DNA stable? Oh yeah, it can stay around for thousands or millions of years theoretically. Because part of it is because it's that double-stranded molecule. The other reason it's so stable is it's wrapped around proteins, right? And then it's condensed. When we talk about gene regulation, we're kind of skipping the chromatin structure part that you guys might have heard in other classes about condensation and melting and all that, heterochromatin and all that. So we're going to talk specifically after that happens, okay? RNA is unstable. Sometimes RNA only stays around for a few seconds. Sometimes it's minutes. Sometimes it's hours. But RNA tends to be very labile. Doesn't stay around for very long. It's a more dynamic molecule. Right? RNA doesn't have to be passed on from one cell to the next. DNA does, but RNA does not. Okay? They're both composed of 4 different bases, or nucleotides. DNA, we know, is thymine, adenine, cytosine, guanine. This is the base portion of the nucleotide, right? We know that thymine base pairs with adenine. What type of interactions hold those together? It's hydrogen bonding, right? That hydrogen bonding between those complementary base pairs is what allows DNA to form that double helix. Cytosine pairs with guanine, hydrogen bonding. Now RNA is different. RNA does not have thymine, it has uracil. That's the different base. Remember that? So uracil hydrogen bonds with adenine. The cytosine and guanine are still the same. Difference in the actual structure is on this ribose sugar in DNA, it's a hydrogen instead of the hydroxyl group. That's another reason RNA is less stable. All basics, right? Now, one thing that we'll talk about more in 329 is RNA can actually form pretty elaborate secondary and tertiary structure. It can form pretty complicated 3-dimensional shapes and structures. An example of that is, you guys last time told me that ribosomes are made partly of RNA. Okay? I mean, that's a single-stranded molecule, but it can form this elaborate structural part of a cell that's used for translation. So even though in here I'll typically draw RNA as just a squiggly line, it sometimes has complex structure to it. Basics there. Here's the genetic code, which we all have memorized, right? Remember, when we talk about a little bit more terminology here, in the nucleus we have, this is a double-stranded DNA molecule. We tend to write it 5' to 3' for the top strand, also known as the sense strand in this case. The other strand is complementary sequence. It's basically the opposite sequence, right? So it runs 3' to 5'. But now we have our A to T and our G to C complementary base pairing across this. The sense strand is the one that has the nucleotide sequence that ends up in the RNA and then the mRNA. So we call that the sense strand. The difference is thymine is replaced with uracil. So for mRNA, we would write this coding strand 5' to 3', changing thymine with uracil across here. Then we translate. So basically this part is kind of the same language, right? It's nucleotide. So we're transcribing it, basically we're rewriting it. In the case of translation, we're changing the language. We're going from nucleotides to amino acids. Now in this case, if we read 5' to 3', we see that AUG is the first codon. Remember, this is read in triplets. Here's AUG, the next one is GCC, and so on. The first amino acid is always methionine in a protein. Second amino acid can be anything. If we look at the genetic code, I want to make just a couple points here. One is there's redundancy in the genetic code. That genetic code has to code for 20 different amino acids. For some of the amino acids, there's several different codons that can cause that amino acid to be incorporated. Remember that? So for example, if we look down here, here's alanine. There's at least 4 different ways that alanine can be coded for. There's a GCU on down. Okay, I'll come back to this in a second. One of the things we're going to talk about today is those interactions between proteins and DNA. So here's the DNA double strand, complementary base pairing between these nucleotides. We can see a major groove, minor groove. Major groove is basically the way the DNA structure is. Proteins can access certain parts of that DNA molecule. Proteins actually kind of reach into the DNA and interact with it through interactions between their amino acids and nucleotides. So the protein-DNA interactions also depend on bonds between specific amino acids and nucleotide bases. For example, in this protein, this amino acid right here, and these proteins would actually could be closer than this. Let's say this amino acid right here, and you can barely see the different colors here. Here's amino acid, here's an amino acid. This amino acid right here is going to base bond, or is going to base pair, is going to hydrogen bond with this particular nucleotide. In other words, if this amino acid was a different one, it might not be able to bond here. If this nucleotide was a different one, it might not be able to form this bond. If all of our DNA is slightly different, that means that these nucleotides could be different between us. That could cause differences between certain genes and certain proteins. Where'd this protein come from? A gene. So if that gene that transcribed and then translated for this protein was slightly different, this amino acid could be different between us. And that could affect the interaction between this protein and a specific gene. Okay, so there are lots of differences between us. Some of them are subtle, some of them are not. One of the things that you heard over and over again in your classes before was a protein's shape determines its function. Remember structure function for proteins? So a protein's shape or structure determines its function. I want to add one more level to that. Sometimes a protein structure determines the level of its ability to function. Sometimes a protein structure determines its level of its ability to function. And I'll give you some examples of that coming up. If that protein's function is to bind DNA, it has to have a specific structure in order to do that. Okay? Some proteins actually require nutrients to bind DNA. So big picture where we're going with this is, how do things from our diet affect protein structure and gene expression? So if we look at the last one, some proteins rely on nutrients that change their structure and allow them to then bind DNA. Okay, we're going to come back to that, and that's how I'm going to show you how vitamin D and calcium are related. Okay. Now, I'm going to start kind of building this. I'm going to expand on this part of the DNA. And I'm going to draw another one that looks kind of like it. So my drawing is going to look kind of like the one up at the top here. Now we're just going to talk about mammalian genes, just eukaryotes. Prokaryotes are different. Double-stranded DNA molecule here. You guys have seen these, right? This is a gene map, basically. What's +1 mean? For what?
First nucleotide.
In? It's going to be transcribed. So this is, +1 is transcription start site. So it says -30, -30 what? Nucleotides. Let's look at our map. Yellow box -30, Tata box. You heard that? What's called Tata box? Because
It's a bunch of
yeah, the Tata element is about, I think it's about typically 8 or 9 nucleotides that has TA repeats. So they call it the Tata box. But actually I think it has TA, T, about 5 A's, a T, and then a few more A's. But it has this Tata box. Okay, so those are the nucleotides that are mostly in here. Let's look at this little gray, I'm sorry, that's brown or tan or something, right? This is promoter proximal element. This is basically we can call this, we'll just call it a promoter element. It's called proximal because it's close to the transcription start site. Each one of these. Why do they have those little rectangles here? That's an element. And we could generically define an element as a short sequence of nucleotides. Why are each of these defined? They're little boxes. What's binding there? Over here. Where are we? We're not really in the we're not in the coding group. This is in the promoter. So what's binding here? Transcription factors. So transcription factors bind promoter elements. So in the research world or in the nerd world of research, when you're doing these kind of experiments, you call this promoter bashing. You're trying to find specific elements within a promoter that are binding proteins. The way you used to do it, you'd use like DNA footprinting and DNase experiments where you can take segments of DNA, you can label them, you can expose them to nuclear extracts, and you can find which parts of this promoter are elements that are binding proteins. Then there was ways that you can actually figure out which protein that is. Okay? So remember, we're in the promoter region of a gene. In other words, these transcription factors are going to be involved in transcription of this particular gene. Okay? We have some other things down here. These are enhancer yeast. This is upstream. Well, let's not worry about this. So don't worry about these. I'll add some more details later. So let's go this way. Down here, what are these? Remember exons and introns? What's the difference in these? What's an exon? You call it an exon because that is a sequence of DNA in a gene that's expressed to determine the amino acid sequence of the protein. So an exon is the part of the gene, is a segment of the gene that is expressed and determines the amino acid sequence of the protein. What's an intron? It's sometimes referred to as an intervening sequence. It's not involved in the amino acid sequence. Okay, so through a gene, through the coding region of a gene, you tend to have a mixture of exons and introns. It goes exon, intron, exon, intron. Again, we're ignoring that green box.
So what's the point of an intron?
Well, now we know it's involved in regulation of translation. Kind of weird. So on a side note, we used to call that junk DNA. Remember that? I think I said that last time. It says junk DNA because we don't know what it does. It must be junk. We're pretty smart. And now a few years ago, the guys actually won the Nobel Prize for this. They realized that intron sequence sometimes codes for a part of the RNA that regulates how much of the mRNA is actually translated. So it's involved in regulation of translation. Parts of it are. Now, let's go through this. One of the questions that we're going to look at is, and you don't have to memorize this, this is just something I took out of a biochemistry text that shows kind of what's happening here. And let me run through this, then I'll show you a little cartoon, then I'll give you step by step on how this works, memory, just because I know this is a review. Here, for example, is the Tata box. So some scientists, a group of scientists that first characterized this, used their imagination. They found a protein that binds to this, and they called that protein Tata binding protein. Pretty imaginative, right? So what's that protein looking for? That Tata sequence. and then it binds it. Okay? That can start kind of a cascade of protein binding. Once TATA-binding protein binds, other transcription factors can start binding. So in this case, we have transcription factor 2B that binds right around that area. Then we have more transcription factors that are binding. Eventually, POL2 comes in. What does that stand for? That's RNA polymerase 2. That's the only one we're going to talk about. Eukaryotes have polymerase 1 and 3 that are involved in tRNA and ribosomal RNA transcription. We're just going to talk about RNA polymerase 2 because that's the one that transcribes for proteins. So that's our business enzyme. So eukaryotes have 3 different RNA polymerases. Prokaryotes tend to have 1. Okay, so we're going to focus on this one. RNA polymerase comes in. RNA polymerase then binds with some other proteins. This is a pre-initiation complex RNA polymerase. And some of these proteins are then modified, and then RNA polymerase starts transcription. So it's a sequence of events. If we look at it on this way, I think this will walk through it. So here's our promoter region of DNA. It's the actual coding region. Transcription factors start assembling on the promoter. Bear with me. It happens faster in a cell. RNA polymerase 2 joins, and then we have transcription. Makes a single-stranded RNA molecule. Okay? Did you guys all watch the transcription video I put up there? Come on, you gotta watch that one. Watch that little transcription movie up there, even if you've seen it before. One of the things that it's showing is, it's showing a strand of DNA, it's showing proteins binding to the DNA, it's showing a specific number of proteins joining that in that region of the promoter, and then it's showing RNA polymerase coming in. Then RNA polymerase is released from those other proteins and starts running through that DNA. As it does that, that RNA polymerase is melting the double strand, it's incorporating the appropriate nucleotide to make the RNA molecule until it runs through to a stop codon. And then RNA polymerase is released. Okay? So when we look at this, we're going to go through the steps now on how this happens. First step is transcription factors assemble together on the promoter. Now in eukaryotes, it's estimated that more than half of all eukaryotic genes have a TATA box. So we can say the first step in most genes is TATA binding protein binding the TATA box. So again, if we look at big picture in a eukaryotic cell, how is this gene found in all of these nucleotides of a chromosome? Well, TATA binding protein is specifically looking for TATA box, and if it's there, it'll bind. So TATA binding protein then helps recruit other transcription factors.
So it'll recruit other transcribing factors?
Transcription factors.
What was the elements?
Yeah, now some of them will bind here. So some of them will bind TATA binding protein. Some of them will actually bind response elements. So TATA binding protein starts the recruitment of other transcription factors. until a general transcription factor complex of approximately 25 to 50 proteins is formed.
General what?
Transcription factors. I'm not going to put them all there. These are general transcription factors. Until a complex of approximately 25 to 50 general transcription factors is formed. RNA polymerase is then recruited. The general transcription factors are required to recruit RNA polymerase because it does not recognize DNA sequences. So the general transcription factors are required to recruit RNA polymerase because it does not recognize DNA sequences. Uh-huh.
Sorry, the RNA polymerase 2 doesn't.
Correct. That's a big difference between eukaryotes and prokaryotes. Prokaryotes, RNA polymerase is looking for specific DNA sequences. In eukaryotes, RNA polymerase 2 requires transcription factors to find specific DNA sequences. So it's recruited by other proteins that have assembled at specific locations of DNA. So a little bit more background information. These guys, some of these transcription factors are looking for specific DNA sequences. Some of these transcription factors are looking for other transcription factors. So when you do these experiments, you end up with some of these proteins. Some of them are not bound to DNA, but they're required. Okay? Some of them are directly involved in recruiting RNA polymerase. So now RNA polymerase is in the proximity. It can now bind to the transcription start site. Once it does that, it can start transcription. Now, can you think of any reason you want to have all these proteins involved?
Oh, you probably don't want it to bind just anywhere because then there'd be chaos.
Specificity, good point. Right, so you need a significant number of proteins. One, they could provide specificity for RNA polymerase. Let's say, for example, any of these proteins kind of make a mistake. Well, they all have to make a mistake in order for polymerase to be recruited. So they help provide specificity. Okay. These are called general transcription factors, right? So let's say, for example, we have a condition where this gene is being expressed at a certain level. Let's say it's being expressed at what's called a baseline level. Or another way of saying that is basal expression. Another way of saying that is constitutive. You heard that term? Constitutive gene expression means basal transcription. It means it's just being transcribed at a consistent level. Genes that might be expressed that way would be maybe cell morphology genes, let's say actin, and some of these genes that are just consistently needed. Now, the requirement for lots of transcription factors can also provide levels of regulation. For example, some transcription factors are actually regulated. And I'll show you an example of this in a second. We have general transcription factors, we have other transcription factors that are also regulated by certain conditions or molecules. So let's say, for example, in this case, we have general transcription factor, general gene expression that's occurring. And let's say, for example, under baseline conditions, we have 2 of these RNA that are being made per minute. Let's say conditions change, so that now we have additional transcription factors. I lost count. Now we have 10 RNA that are made per minute rather than 2. That can happen if more of these transcription factors are on the promoter region and help recruit RNA polymerase. Okay, and I'll give you an example. I really got to get rid of that. I'll give you an example in a few minutes. Let's go through this first. Now, once we have transcription that takes place, right? We have all these RNA molecules. They are full-length molecules relative to the gene. So those are called, I generally just call them RNA molecules. Sometimes they're referred to as nuclear RNA. This is where the DNA, basically the gene has been transcribed. It still contains the intron sequences. The introns are spliced. The exons are joined. And the RNA converts to mRNA. So after transcription, splicing occurs. Introns are removed. That results in an mRNA. Its sequence is changed and it's shorter. Where does this occur in the cell? The nucleus. Then the mRNA is exported into the cytosol. So this is splicing. Equals the introns are removed. Another, well, the category that this falls under is this is the type of RNA processing. The RNA is processed. So the introns are removed. Now you have mRNA that's exported into the cytosol for translation to start. So in the cytosol is where the ribosome binds for translation. So let's look at this. This is where our RNA has been made. The mRNA has been exported into the cytosol. Now translation starts. Remember, that's where the ribosome assembles. Translation. You guys learn about it. What? Biochemistry? Bio 161? Okay. So we're all up to speed on this. All right, remember the small subunit of the ribosome binds mRNA. The initiating tRNA binds. Large subunit of the ribosome assembles. Translation elongation starts. That's when the tRNAs are entering the top of the ribosome. The tRNA anticodon sequence pairs with the codon of the mRNA. What kind of bonding? Hydrogen bonding. What's this? That's the amino acid, right? These tRNAs are charged, remember that? If the tRNA is charged, that means it has an amino acid. So tRNA, a charged tRNA comes in, base pairing occurs. If it's a wrong tRNA, it's kicked out. Next one comes in, right? Until it's complementary base pairing. Then what happens? Peptide bond is formed. This amino acid is added right here. So peptide bonds are formed. Where does that occur? In the ribosome. So peptide bonds are formed within the ribosome. And a peptide / protein is made. So this ribosome is going to keep moving. It's actually pulling this mRNA through until it gets all the way to the end. It'll find a stop codon, and then translation stops, thing disassembles. Pretty basic. But you want to have those details in mind because the next thing that we're going to talk about kind of requires those details. Right? Remember what's happening here is this peptide is actually coming out of the top of the ribosome. tRNAs are coming in, peptide bonds are formed, peptide gets longer and longer. It's coming out the top of the ribosome. Another way of looking at it, that's all. I'll let you read through that. This is really basic. There's lots of. I used to have like little tutorials and stuff on the PowerPoint. Nobody's looking at them, so if you want to find those, you can. Review your notes, whatever you need to do on translation. But basically, this is what happens. We get a protein strand that's made here. A couple more points, and I'm going to backtrack. Let's say, for example, in DNA, a one nucleotide change or mutation is at the DNA level. You could look at this as, for example, a somatic mutation, say in the case of cancer, or you could look at it as my DNA being one nucleotide different than yours. Let's say I have a gene that has a single nucleotide that's different than yours. That can lead to a change in the nucleotide at the RNA. That will. That can lead to a change in the protein structure. Classic example is sickle cell anemia. Remember that? I'm going to give you another example in a second. But in this case, sickle cell anemia decreases oxygen carrying ability of hemoglobin, causes medical problems such as nicking small blood vessels, lots of physical abnormalities and complications because of that. The hemoglobin actually becomes abnormal. And rather than being kind of globular tetramer of protein, those hemoglobin molecules can actually link to each other. They kind of stick to each other. They form these long fibrous proteins, and that distorts the red blood cell shape. Because of that, that red blood cell loses its physical flexibility, can't move through certain blood vessels, and that causes a lot of abnormalities. In addition to that, that hemoglobin is not binding oxygen the way it should. All of that's caused because of a single nucleotide change. But in this case, normal hemoglobin has a glutamate. This is a side chain of glutamate. CH2, CH2, carboxylic acid group. That can be changed to a valine. Valine is a hydrophobic amino acid. This is hydrophilic, this is hydrophobic. Here's the side chain of valine. That's one of the branch chain amino acids. Okay? And that's kind of what I just talked about. Get that sickle cell classic morphology. This is what we're going to talk about next. Nutrients can actually affect a gene's expression. I think there's a typo in yours. This should be an A. Sorry. I read through that. I did it again. So that's got to be an A. Nutrients can actually affect a gene's expression by either increasing or decreasing a protein that's being made. So here's an example. Vitamin D increases the expression of a protein that's needed for the absorption of calcium. In order to go through that, I'm going to use this little insert right here. And the diagram I already have up on the board. I'd rather run through this kind of in one step, so let's take our break right now and then we'll come back and I'll start this. Alright, so hopefully this is recording again. It looks kind of weird. I'm not quite sure why. It should be. Let's see, where were we? That looks like something useful. Alright. Okay, let's add in a little bit more to that diagram I have up there. Question I asked you last time is, why do they fortify milk with vitamin D? And you said because it has calcium. You want to elaborate? Why is the vitamin D important? Helps absorption of calcium. How?
Genes.
I just like that. Because don't people pressure you, and you're like, I'm a nutrition major. Oh, I'm going to ask you. So what about this? And you're like, yeah, because of the. Well, how does that work? I don't know. So a lot of the stuff that we're going to try to go to the level of how things work in here. So you should be able to kind of look at it like, not only should you know what time it is, you need to know how the clock works. Okay? Not enough just to say, it's 5 o'clock. But how did it actually get there? Okay, so how does vitamin D actually work to regulate calcium absorption? And what do you know about vitamin D? What kind of molecule is it? It's a vitamin. Is it? It's fat soluble. It's a hormone. We can make vitamin D in general. We make it from cholesterol in the skin, so when you go outside in general, that sun exposure because of the UV light helps convert kind of cholesterol metabolites eventually to a form of vitamin D. It then goes into the bloodstream, then it has to go to the kidneys, has to go to the liver for activation. Then you get this vitamin D molecule that works as a hormone. And what's the definition of a hormone? Molecule that's some of them, so you got a little bit. A hormone is a molecule made in one part of the body that can elicit an effect in a distal part. Remember that? Made in one part of the body, travels to the blood, to another part of the body to cause effects. Vitamin D can be made in the skin, travels through the blood. Where do you think in this case its effects are relevant? Bone's important. What else? Milk. Part of the digestive system. Intestinal cells. Okay, so look at it this way. This is an intestinal cell. More specifically, An enterocyte. What does entero mean? The gut. Cyte is a cell. So this is a cell in your small intestine. Slightly smaller in your intestine. What's this? Brush border membrane, yeah. Right, so this is brush border membrane. This is basolateral membrane. Remember that term? Some of you haven't had that yet. Basolateral membrane. So what's over here? This is the blood. Specifically, that's going to go in the portal vein. That's going to go to the liver. So this is the blood. So this is the opening of the small intestine. So we're going to call this the lumen of small intestine. And here is our star that we're going to talk about, calcium. In this case, what do we say it's from? Milk. Okay? Now, there can also be vitamin D in there, right? Typically there is in the U.S. That vitamin D can actually go through these intestinal cells into the blood, then it can go to the liver, then the kidney, then it's activated, then it kind of comes back to the enterocyte. So that vitamin D that you consume with one particular glass of milk might not help until the next time you consume calcium. But if it's in there consistently and you're consistently drinking milk, it'll have a consistent overall increase in calcium absorption. Here's how it works. We look at this inset right here. Okay? A little bit of the terminology. VDR, so you want to write this down. VDR stands for vitamin D receptor. VDR is vitamin D receptor. What kind of molecule is it? It's protein. This whole box right here is representing a cell. So this is basically cell membrane. This is plasma membrane. So VDR is in the cytosol. This part of the cell is this. What does that look like? DNA. This is the nucleus. So label this as nucleus. See where it says D3 right there? That's vitamin D. We're assuming already activated. Vitamin D. Yeah, there's vitamin D2, vitamin D3. D3 is from animal products, and we make D3. D2 is from plants. We'll talk about that next quarter. So for now, we can say vitamin D. Look what happens. VDR can enter the cytosol, the nucleus, from the cytosol. Vitamin D comes into the cell, goes into the nucleus, binds VDR. So in the nucleus, vitamin D binds VDR. Anything different about VDR there? Change in shape. VDR goes into the nucleus, binds vitamin D, VDR changes shape. VDR then binds RXR. This guy right here. I'll tell you what that stands for. You might not have known this. Retinoid X receptor. When you hear retinoids, what do you think of? Vitamin A. Retinoids are a class of vitamin A molecules. This was called retinoid X, kind of a funny story, because when they first found this protein, they didn't know what it bound. It was what they call an orphan receptor. Oh, we know it's a receptor, we don't know what it binds. They eventually found out its high affinity ligand is 9-cis retinoic acid. For us, though, we can say, they just said, oh, it's some kind of retinoid, we'll call it retinoid X. For now, just remember RXR is retinoid X receptor. We'll talk about it now and later on as well. After that, VDR is then phosphorylated. See these little P's here? That means amino acids of VDR have phosphates attached. So VDR is then phosphorylated. VDR then binds VDRE. This is alphabet soup today. VDRE, which stands for the vitamin D responsive element. What did we say an element is? Yeah, a short sequence of DNA. In this case, what's it related to? Vitamin D. In other words, this is a response element that actually responds to vitamin D. So here's how it works. Vitamin D goes into the nucleus, binds VDR, that then binds RXR, phosphorylated. VDR then binds a vitamin D response element. See this sequence right here? That's what's called the consensus VDRE sequence. In other words, this vitamin D responsive element is made to bind VDR. How is it bind? How is it binding? Hydrogen bonding between VDR amino acids and these nucleotides. So this is sequence specific. VDR looks for VDRE. Between the amino acids of VDR and the bases or nucleotides of VDRE. Now this is a regulated response element. It's not a general response element. It's not a general element like these guys that we said up here. I remember we said general transcription factors tend to bind pretty close to the transcription start site, recruit RNA polymerase. Now, for example, we're looking up here a little bit. I'm going to put this in like this. Let's say this is VDR. This is vitamin D. So what would this be? Here's the VDRE that has a sequence that looks similar to that. So now let's think about what happens is, we said that these transcription factors can help recruit or can affect the recruitment of RNA polymerase. In this particular case, what we're going to say is, if vitamin D levels go up, we have more VDR that binds to VDRE in specific genes. VDR can increase the recruitment of RNA polymerase. VDR can help increase the recruitment of RNA polymerase. So in this case, what do you have? RNA polymerase is recruited more efficiently, so you have enhanced transcription. We have enhanced transcription because we have more vitamin D, more VDR is now in its active form, binds here, recruits RNA polymerase, increased transcription. So we have enhanced transcription of this particular gene.
Is it only one particular gene that helps transcribe, or is there multiple genes that could be a transcription factor?
Multiple. So good question. Everyone got this so far? Now,
Why does it increase the recruit? Is it just because?
in this case, it's somehow helping to recruit some of these other transcription factors that RNA polymerase is recognizing. So now one thing to keep in mind is this. In a lot of cases, gene expression is not yes or no. It's relative levels. So we can't look at it like the gene is either on or off in this case. This particular gene is constitutively expressed. Some is always being made because this particular gene product is required for the cell to survive. But in this case, if there's vitamin D, transcription goes up significantly. So we might go from 10 transcripts per minute, from 2 to 10. So let's say, for example, I'm consuming more vitamin D, gene expression of this goes up. If my vitamin D goes down, gene expression goes down. So the gene product goes down. In this case, what does that mean? Calcium absorption goes down.
So is this like if you build stronger bones or like your hair grows longer or something, that's like the genes being expressed for those things because not saying that's what vitamin D leads to, but say if it did lead to those things. Or like when you take certain vitamins for things, is that what's going on? Is it
In this case, yeah.
okay.
In this case, we have certain genes that have the ability to be regulated by vitamin D. Certain genes. Now, so Aiden asked a good question. So is this what genes does this involve? Only the genes with VDREs. This particular gene has VDRE sequence in its promoter. Not all genes have that. Now, what's interesting about vitamin D is, and I'll tell you a really short story about this. When I was in graduate school, the guy that figured this out, his name is Mark Hausler, and he was actually a biochemist, and he was in the medical sciences part of our university. I went to talk one day, and the guy walks in, and I thought, well, I don't know who that guy is, but that guy looks like some kind of rock star. When's the scientist coming in? So the guy walks in, it's hilarious. Very, very arrogant. Walks in, this and that, I'm just like, oh man. And then the more he started talking, I was like, that's really cool, no wonder he's arrogant. He was one of the first guys to figure out how a nutrient regulates gene expression. It was vitamin D. So what they did in their lab was they started finding that certain gene products are regulated by the presence of vitamin D. So what did they do? Promoter bashing. And they figured out that this particular sequence was the one that was involved in vitamin D regulated gene expression. In other words, when they did recombinant DNA type of experiments, I won't bore you with the details, they would take genes that were regulated by vitamin D, and then they'd start knocking out specific nucleotides here, and they could see that it affected vitamin D regulated gene expression. Okay? So then what they started doing, then you start expanding and seeing which genes have VDRE. You know what they found out? Lots of them. That's when they went, oh, vitamin D is not just involved in calcium absorption, it's involved in a few hundred genes. Some of them are related to the immune system, some of them are related to cell division, some of them are related to protecting against cancer. So in other words, if you're working with a gene and you backtrack and you start looking at the promoter sequence and you see VDRE, what does that tell you? That gene's probably regulated by vitamin D. And there's lots of them like that. Okay? The most famous one is the one we're going to talk about right now. Okay? So we're going to give this a name. The protein that's made from this is a protein that does this, and I'm going to, you guys know this, calcium's charged, so it needs a transport mechanism to go across the plasma membrane. Once it goes in, this is what happens. Calcium binds this protein. This protein moves calcium through the cytosol of enterocytes. So in general, the more of this protein you have, the higher calcium absorption. Guess the name of that protein. Calbindin. So we're calling this the calbindin gene. The more vitamin D you have, the more VDR is activated. The more VDR molecules can bind to VDRs in the calbindin gene can increase efficiency of transcription. More transcription, more RNA, more mRNA, more potential translation in the cytosol. In general, the more mRNA you have, in general, the more translation can take place. Okay? So for example, if we looked at this cell in more detail, we could have the nucleus right here. Say this is the nucleus. Here's our calbindin gene. Right? VDR, we'd have vitamin D here. Let's say we have vitamin D right here. It's bound to VDR. And that's driving all of this. So then what happens? The mRNA comes out. Then you have protein. You have translation. To calbindin protein. It's not a very good diagram, but get the point. And it can cause a pretty significant effect. It can, boy, I'll give you numbers later, but vitamin D has a dramatic effect on calbindin levels, so that can significantly change the levels of calcium absorption efficiency. So if you lack in vitamin D, it can decrease calcium available for things like bone formation and other things that calcium is required for in the body. Does that make sense? So in this case, what's happening, vitamin D is kind of an on-off switch for this. It's regulating the amount of calbindin that's actually made.
What's the annotation? It looks like NC.
Nucleus. Simple? Okay. Remember, when that vitamin D binds VDR, it changes the shape, and that's what kind of starts this process. It's all about vitamin D binding and changing the shape. If you want, next time I'll show you. I've got a three-dimensional structure of this. I can show you how vitamin D is actually bound in there. Good? Okay. Let's go to the next topic. Now, so far I think in your other classes you should have a pretty good handle on translation. And I think what's interesting to me is a lot of times translation is talked about in the cytosol and it's just left there. When in reality, probably about 2/3 of cellular proteins don't stay in the cytosol. They either go to other intracellular organelles or they're secreted from the cell. So you can make an argument that the most relevant mechanism of translation is the secretory pathway of translated proteins. So we're going to go kind of one step further based on what you've learned so far. So it says not all newly synthesized proteins remain and function in the cytosol. Some proteins need to be transported to the nucleus, mitochondria, or lysosome, while others are secreted from a cell. For example, livers make a protein known as albumin. Have you heard that? Did you guys do a blood sample and run it on a gel in biochemistry? Or did you look at it or something? You did saliva and IgA, that kind of stuff? Okay. If we took a drop of your blood, we delipidated it, and then we put it on an STS page gel, and we ran it out there, we'd see hundreds of proteins. You'd see a few proteins that are extremely common. You'd see really significant dark bands. Probably the three most common would be heavy chain and light chain of the IgGs, of the immunoglobulins, sorry. The other one, albumin. It's a significantly high level of protein in the blood. So liver cells make that. Liver cells make albumin and put it into the blood. Albumin performs a variety of functions. One of its functions, for example, is it transports fatty acids in the blood. So we've talked a little bit about exercise in here, and we said, for example, fat cells can actually put fatty acids into the blood, and then other cells can use that for energy. How does the fatty acid get from the fat cell to the exercising muscle fiber? Most of it is attached to albumin. So albumin can transport fatty acids through the blood. How did the albumin get into the blood? That's what we're going to learn. The secretory pathway of proteins begins as a protein is targeted to the ER for co-translational insertion. That's a term I made up. Hopefully that's not been patented or anything. Into the inside of the ER, you'll see what that means in a second. After translation, segments of the ER bud off and translocate to the Golgi for further processing and preparation for export. A little bit more about cell biology before we move on. Here's the nucleus, nuclear envelope. One of the things we said was on the outside of the nucleus, pretty common for that nucleus to be surrounded by the ER. A lot of that ER is containing ribosomes, so we know that's called the rough ER. These little dots here are supposed to represent ribosomes. What are they doing there? We've all seen that, right? Oh, the RER. Why are they there? Well, ribosomes do translation. What we're going to see is they actually translate proteins and insert them into the RER. They go inside. They don't go into the cytosol. Okay? So the ER, what is it? Well, it's a network of membranous channels in the cytoplast that links the nucleus, the Golgi, and the plasma membrane. In other words, proteins can move through the ER to get to the plasma membrane. It is important for the synthesis of membrane lipids and proteins. The difference between the ER and the RER are ribosomes. The RER is more abundant in cells with high protein synthesis, like liver cells. The majority of the ER is RER. That is the location of most cellular protein synthesis, is on the surface of the RER, not the cytosol. Most secretory proteins are those destined for other organelles, so we'll look at some examples coming up. We're going to spend a little bit of time on this diagram. This diagram is no longer in your text. So when I reviewed one edition of the text, I said, make sure you leave this diagram there, because that's really important. The next edition didn't have it, so they basically said, there's what you are. That's how high you are on the food chain. So it's so important, though. I want to go through this and give you all the details that you need for this. If we take a step back, where are we? This is the lipid bilayer that is composing the RER membrane. So you kind of look at it like this way if you want. This would be enclosed here, and this would be the RER. Because it's the RER, that means there are ribosomes on its surface. So these little double circles here, that's a ribosome. So what would we call this? You can call this the RER cisterna, the inside part, or you could call it the RER lumen. This is the inside of the RER. Let's look at these bullet points before we do more labeling. Now, as the protein is synthesized, it's inserted into the RER. This is not the case for all proteins. Some are translated by free ribosomes into cytosol. That's what you guys have mostly learned. Translation into cytosol. Let's identify a few more things. See this long line right here? That's supposed to represent an mRNA. They're trying to show this sequentially. So another way of looking at it is a little short mRNA right here. They're just trying to show it moving along in this sequence, A to G. So this is the cytosol. Here's translation starting. Small subunit, large subunit, what's this?
The amino acid chain.
That's the peptide. That's right here. Okay. So here's where we're going to start. So let's identify these. SRP equals signal recognition protein. SRP equals signal recognition protein. DP, docking protein. You ever heard of that? Docking protein. SP, signal peptide. SPase, signal peptidase. I think that's it. You know the players. Let's go through the game here. Translation starts in the cytosol. Step 1. Translation starts in the cytosol. Blood glucose is low. Hang with me. A signal peptide extends from the ribosome. In general signal, these signal peptides are about 25 to 35 amino acids long. The top of the ribosome. The signal peptide is bound by what? What recognizes it? Signal recognition protein, that's its job. Finds that signal sequence. So the signal peptide is bound by SRP. SRP moves the translation complex to the surface of the RAR.
Is that complex just like the signal peptide?
Good question. What is the complex? The translation complex is mRNA, ribosome, SP, I guess you could say SRP as well. So that whole complex moves to the surface of the RAR. SRP helps insert SP through a channel protein. It's hard to write and visualize at the same time. I just kind of, I don't know what's going on. So this is what's going on. SRP binds. What's it looking for? It's looking for a specific amino acid sequence. This sequence relatively uniform. In other words, this SRP is looking for a specific amino acid sequence. If it's very far from that, SRP won't bind. Okay? SRP binds it, moves the translation complex here, helps insert the signal peptide through this channel protein. SRP is then released. It has to be released because that little peptide has to go through there. SRP doesn't. So SRP is released and can repeat the process. That's what this means. SRP is released, can repeat the process. Docking protein then stabilizes the ribosome. Co-translational insertion begins. So as the name implies, what's happening? As it's being translated, it's inserted. Inserted where? Lumen of the RER. Where's the mRNA? Cytosol. Where's the ribosome? Cytosol. Doesn't go in the RER. Don't put your mRNA in the RER. Okay, so here we are. As the signal peptide enters the lumen, it's cleaved. As the signal peptide, so as SP enters the lumen, it's removed by signal peptidase. That's what this is showing. Signal peptidase, cutting signal peptides. So these 35 amino acids removed. Translation continues. Until completed. So translation continues until it's completed. And the ribosome disassembles. So where's the protein? It's in the lumen of the RER. We've got a completed protein that's now inside the RER. So if we look at it this way, where are we talking about? That would be happening like right here, for example. An mRNA molecule would be translated. You can see the ribosome. They always show that. They don't show the channel protein, but what's happening is the protein's actually being inserted in here. So now you have a completed protein that's been translated. It's inside the RER. Why is it in there? So now, because it's going to be transported to different places. Okay? So the completed protein can be folded. And then transported to the Golgi. And we'll kind of continue on with that 1 second. Yep. Yeah, it would be cleaved into amino acids. Those could be used for other things, yeah.
If your body doesn't want to ship it off, will it cleave it early or will it just not even process it when it's during transcription?
doesn't want to ship it off. If that protein needs to go someplace utilizing the secretory pathway, it'll have a signal peptide that performs that function. If a protein does not need to go through the secretory pathway, it will not have that signal peptide.
Okay.
So let me put it this way. There's a few of these. What I could do in the lab would be this. I could take any protein. I could use recombinant DNA technology. I could alter a DNA sequence. I could put that DNA sequence into cells. I could translate that protein. I could make any protein go through this, as long as I add what to it? Signal peptide. So lots of proteins have the signal peptide because they're going through the secretory pathway, but a lot of proteins don't. So if you think about it, what's an example of a protein that would just need to stay in the cytosol, perform function in the cytosol of any cell type? You think of any proteins?
The protein that combines with RNA to make the ribosomes.
Oh, it could be some of that, yeah.
Pyruvate dehydrogenase?
Yeah, could.
Would that have to be shipped to the mitochondria?
Which one was it?
Pyruvate dehydrogenase?
That would go into the mitochondrial matrix. Any other proteins in the cytosol of any cell type?
The proteins that DNA wraps around to
Those would be in the nucleus.
it's just
Glycolysis enzymes?
Glycolytic enzymes. Sometimes in life it's right in front of you, ladies and gentlemen. How about calbindin? Cytosol of enterocytes, right? So calbindin wouldn't go through this. It functions in the cytosol. Would the calbindin gene need to have a signal peptide as part of it? No. Not going through the secretory pathway, staying in the cytosol. Glycolytic enzymes, glucokinase, any of those guys, stay in the cytosol. So that would be the translation you learned before. Translation cytosol, protein stays in the cytosol. If a protein is going to go through the secretory pathway, it's got the signal peptide.
Alright, what exactly is the secretory pathway?
The secretory pathway is basically this. Proteins being translated, going through the RER, through the Golgi, and then ending up in different parts of the cell. Secretory pathway doesn't mean that they're all secreted. It means that they are not staying in the cytosol. So they can be secreted, they can be embedded in the plasma membrane, or they can go to other organelles.
Are there any kind of proteins that can be used in the cytosol and or be shipped off? So there's like an on-off signal protein sequence for that gene?
There could be. Yeah, there's some things that regulate where they finally end up in the cell or if they're secreted. It would maybe be a different mechanism other than the signal peptide. Yeah. Yeah, there's some enzymes that are related to that.
During this process, you said the mRNA and then one other compound stays in the cytosol. I don't remember which one.
Ribosome. Ribosome's right there on the surface. What does N stand for?
N-terminus?
That's the N-terminus. So this is the first part of the protein that was translated. So the first part of the protein that's translated, signal peptide. That's kind of what cues everything. Okay. The Golgi. Now, have you talked about Golgi and other things, classes before? So for us, we want to keep in mind one important thing for the Golgi, and that's glycosylation. Glycosylation, that's the addition of carbohydrate. And that addition of carbohydrate or glycosylation can help a protein perform its function. There's lots of proteins that are glycosylated. Remember we talked about plasma membrane? We said, you know, there's lots of proteins in there that actually have carbohydrate attached. How did the carbohydrate actually get there? The Golgi. Like these guys. These proteins, carbohydrate attached. Guess how these proteins got to the plasma membrane? secretory pathway. That's how they got there. Remember GLUT? Transports glucose, the plasma membrane, integral membrane protein. How did it get there? Secretory pathway. Insulin receptor? Where does it end up? Plasma membrane. What cell type? We talk about muscle cells. Also fat cells. Insulin receptor embeds in that plasma membrane. How to get there? Secretory pathway. Goes right along this process. Okay, so we'll look at some more examples coming up. So the Golgi is the next step of protein sorting and trafficking for secreted proteins after the RER. The Golgi is composed of membranes, flattened vesicles. Proteins move through the Golgi in a stepwise fashion by budding off and fusing with the membrane of their target. I'll show you that in a second. The cis Golgi network accepts newly synthesized proteins from the RER or the ER. So it goes cis to trans Golgi. Trans Golgi is the exit site from the Golgi. Major functions of the Golgi. For us, if you think Golgi, think glycosylation. So the polysaccharides are added, and then they can be inserted into membranes. It's responsible, along with the ER, for synthesizing membrane organelles and the plasma membrane. The ER and Golgi comprise the secretory pathway for proteins. And this is kind of what we talked about, or I showed you. Let me show you these three examples real quick. This protein, for example, in pathway 1, has gone through the secretory pathway, through the RER, through the Golgi, and is now being exocytosed. What's an example of one of those proteins? Albumin. Albumin from liver cells. Could be this guy, into the blood. This protein has gone through this. It needs to end up in the membrane. So what's a couple examples for this? It'll actually stay in the membrane. GLUT, insulin receptor, has gone through, stays right here. It's hard to see, but that's what those are showing. Integral membrane proteins. This one over here, pathway 3, some proteins actually go through the secretory pathway and end up in other organelles. For example, lysosomal enzymes go through that process. Okay, so we went through this today. This should be on YouTube. Does that seem like a lot of info today? Gene expression, vitamin D, secretory pathway, a few examples. This should be on YouTube, but look through your notes. And then next time, if we have questions on this, we'll go back through it, and then we'll start digestive system. Yes? On which one? On transcription? Transcription is on, I have that little movie posted on Polylearn. Yeah. That's what this is showing. Lysosomal enzymes, how do they get into the lysosome? Secretory pathway. This is a lysosome. I have to convert this, and then once I convert it, turn it into a YouTube, I'll send you the link. So you'll get an email as soon as I do that. Okay, guys, thank you. Have a good night. It is 6:01.
Does the Golgi also do, I guess it'd be called like oscillation?
It does.
It does.
Yes. Yep.
Alright, see you guys later.
Okay.